So dear student, very good morning to you all. I welcome you all into the industrial pharmacy first. Dear student, we had started the parenteral products, and in our very last lecture, we had discussed the details associated with the process of manufacturing, the plant layout, and design. So let's have the summary of the things that we had already covered in our previous lecture. So we had covered the concept of parenteral. So this is till so far what we had covered, right? And in the very last lecture, we had gone through the plant layout for the production of the sterile preparations. Now, today we have to go through the different aspects associated with the pyrogens. And after going through, after understanding, after studying all these details, you will going to able to explain, you will understand that what the pyrogens are, what is meaning of pyrogenicity, what are the endotoxins, what is its structure, what will be the sources of contamination, and how we can go for process of elimination of the pyrogens. And last but not the least, the process by which we can go for evaluation of the parenteral products. So in order to understand all these, our lecture plan that will move through the introduction to the pyrogens and its importance, the endotoxins, their classification, their structure, the bioactivity of the endotoxins, the different sources of pyrogen contamination, how we will go for eliminating the pyrogens, and last but not the least, that how we will go for the process of quality control for the parenteral products. Now, as far as the pyrogens are concerned, what these are basically the bacterial endotoxins. These are going to be produced by the microorganisms. These are their metabolite product, which are said to be the pyrogens. And when the pyrogens, they will appear into the parenteral product, that is not desirable, because it is the strict ideal requirement for the parenteral product that it should be non-pyrogenic. So such pyrogen contaminated products that are not at all desired, that are not at all accepted. Now, when they will remains into the parenteral product and when it will goes into the body, it will shows a series of reactions and its effect in the form of, say, rise into the body temperature, that is the fever, there is a redness, there is a pain, that is said to be the swelling and known popularly as a inflammation. There is a fall into the blood pressure, that is the low blood pressure, that will lead to the process of shock conditions. It will lead to the process of hemorrhages, thrombosis, sepsis, and the tissue damage. So all these kinds of the effects that are caused by the pyrogens when they will enters into the body. When these number of severe, mild to severe effects of pyrogens are there, then the presence of such agents that is not at all desired in case of the parenteral product. Now, basically, if we see what the pyrogens are, these are the lipopolysaccharide. So, lipopolysaccharide, these are the complex of pyrogenic lipopolysaccharide. It is a protein product and it is present in case of the lipids. Now, the bacteria, if we see the bacteria, they can be classified as gram negative and gram positive bacteria in case of the prokaryotic organisms, and based on their behavior against the different staining methods. Let's say, for example, the gram staining. So based on the gram staining and showing the color to that of the, or showing response to that of the gram staining, they had been classified as gram negative and gram positive. Now, What the gram-positive bacteria are? These are the bacteria which will possess the thick mesh-like cell wall, which will be consisting mainly of the peptidoglycan membrane and that surrounds the cytoplasmic membrane. So this is the cell wall of the gram-positive bacteria. You can see that they will contain the different components like say the teichoic acid, lipoteichoic acid, there is a peptidoglycan membrane, there is a NAM, there is a NAG peptides, and there is a cell membrane. So the different examples of the gram-positive bacteria are Streptococcus species, which is responsible for producing the pneumonia, that is the infection and inflammation of the lungs. Then there is a Bacillus anthracis, which is responsible for causing anthrax. And there is a Clostridium species, which is responsible for producing the botulinum and the tetanus causing bacteria. As far as the gram-negative bacteria are concerned, their examples are Vibrios, Salmonellas, Shigella, and the E. coli species. Now, if we see the basic differences, the cell wall is more complex compared to that of the gram-positive. The peptidoglycan layer is much thinner and an outer membrane that can be found on its external surface. So the structure of endotoxin, if we see the lipopolysaccharide, what are their properties? These are heat stable. These are resistant to the proteolytic enzyme. They cannot be degraded by the proteolytic enzyme. These are immunogenic. When they will enter into the body, they will show the immunological reaction from mild to moderate. They cannot be converted to the toxoids and are produced by which bacteria? These are produced by the gram-negative bacteria. They will exist in high molecular weight aggregate form. The monomer unit of lipopolysaccharide is less than 10,000 Daltons and enabling endotoxin to easily pass through the sterilizing 0.2 micron filters. So even a normal process of filtration that cannot go for removal of the pyrogens. So once they will get contaminated into the product, it is very, very difficult to remove them. So the strict care that is necessary to be taken during the process of production of the parenteral product, that there should not be any kind of contamination by any way at any stage during the compounding and manufacturing of the parenteral product. Now, if we'll see the different cores or the different structures that are present in case of the lipopolysaccharide, it will shows the lipid A portion, the R core, and the O specific side chain. The lipid A part, that is hydrophobic structure, and it is showing the toxic biological effects. It is a specific glycophospholipid, usually a bisphosphorylated disaccharide backbone acylated with fatty acid chains. The R core, that will show two different core, the inner core and the outer core. The inner core, that is the same composition in all gram-negative bacteria, and it will contain rarely occurring components like say the heptose, hep, and 3-deoxy D-mannose 2-octulonic acid, and is linked to the lipid A part. Whereas the outer core, that is made up of 6 hexose component and is linked to the O specific side chain. The outermost element of lipopolysaccharide, that is hydrophilic O specific side chain, which is built up from 1 to 40 repeating units of 2 to 7 sugar components. So if we see the bioactivity of endotoxins, the toxicity of lipopolysaccharide, that is associated with the lipid A part. However, most of the toxicity is brought about by mediators. Now, instead of the lipopolysaccharides directly destroying the host cells and tissues, the bioactivity of endotoxins, that will depends on its concentration. So the macrophages, they will induce the production of the interleukins, different kinds of interleukin, like say interleukin 1, interleukin 6, interleukin 8, TNF, that is tissue necrosis factor alpha, and interferon gamma, etc. So these are the macrophages. They will has the receptors onto their surface, and when the antibody bind to these receptors through their Fc portion, and the antigen that is in contact with that of the lipopolysaccharide component of the bacteria, at that time they will causes the release of the cytokines, and these cytokines are responsible for producing the immune responses. So the different kinds of the cytokines like say interleukin, prostaglandins, leukotrienes, these are responsible for powerful inflammation and septic shock causing mediators and are required for the adaptive immune response. So it is very important not to have the uh, pyrogens in case of the product. The characteristics of the endotoxic shock, that is in the form of production of the hypotension, disturbances of the organ function, metabolic acidosis, thrombocytolysis, and the disseminated intravascular coagulation, that is the DIC. So what will be the sources of pyrogen? All, like say right from apparatus, medicaments, solvents, raw materials, stages, equipments, everything that will going to be acting as a source of contamination. So that's why it has been said that, that's why it has been told that we should keep everything that is in sterile condition, and not only in sterile condition but it should be free from that of the pyrogen, because once the pyrogen they will get contaminated, they even cannot get removed through the process of filtration method. So that's why it is very necessary to take care at each and every stage. Now, what are the method by which we can go for removal or elimination of the pyrogens? So here are the methods, like say adsorption method, adsorption on the surface of the selective adsorbents. The different agents can be used as an adsorbent. Then the heating, the solvent extraction, heating at high temperature of 250 degrees Celsius for 45 minutes and exposure of 650 degrees Celsius for 1 minute, or exposure at 180 degrees Celsius for 4 hours. You can see the relationship. As we will go on increasing the temperature, the duration of exposure that will goes on in decreasing. As we will go on increasing the duration, the temperature required that is less. There is an inverse relationship. At 650 degrees Celsius, just 1 minute, that is sufficient in order to eliminate the pyrogens. If we will reduce the temperature from 650 degree to 250 degrees Celsius, we have to heat it up to 45 minutes. And still, if we will go on reducing the temperature from 250 to 180 degrees Celsius, we have to heat it for the process in order to remove the pyrogens for a duration of 4 hours. You can see the temperature which is required, that is very very high. Even in case of autoclave, how much temperature we are going to be maintaining? So that is at 121 degrees Celsius. At max, in case of the hot air on, we can go for sterilizing the product at 160 degrees Celsius for just 2 hours, isn't it? But in order to remove the pyrogen, we have to heat it. The minimum temperature is the 180 degrees Celsius, and that too for the 4 hours of duration. So the process of sterilization that cannot be uh, responsible for the removal of the for the elimination of the pyrogens, or the simple sterilization process cannot remove the pyrogens. So we have to go for some different alternative ways in order to remove the pyrogens. Then there is a selective extraction procedure, like say the careful heating with the dilute alkalis, dilute acids, or mild oxidizing agents in order to eliminate the pyrogens, and the ultrafiltration. The ultrafiltration membrane that is going to be used, and the solution that will be passed through the ultrafiltration membrane, so that the pyrogen they cannot be passed and they can be eliminated. They can be filtered, but that too under pressure only. So we need to control the pyrogens at both the stages, at compounding stage as well as at the manufacturing stage, right? So we need to require the careful planning. We need to require the process of completion As soon as we are starting on the same day, we should carry out the process of compounding and manufacturing. There is no uh, taking over of compounded product, or compounding that could be carried out on one day and manufacturing that could be carried out on another day. No, it is not like that. On the same day, the product that should be compounded, and on the same day it should be manufactured, filled, sealed, right? Because in order to avoid the process of contamination. So the manufacturing that will involve the process of planning on the same day, completion on the same day, and the process of sterilization. So expeditiously within the maximum allowed time, or on the same day itself. The manufacturing process that must be rapid, right, in order to avoid the pyrogen contamination with no risk of microbial contamination. The manufacturing should be within one working day, including sterilization. Now, once this product is manufactured, that is say the parenteral product, it has to subject to the process of quality control. Now, quality control, that is nothing but ensuring that the product that is free from that of the pyrogens. Now, in case of parenteral product, not only we should ensure the product that is free from that of the pyrogen, but it is sterile, it is free from that of the particulate matter, and it will not contain any kind of leakages into the container, so that the contamination that will leads to. Now, as far as for the process of quality control or the evaluation of the parenteral product for the pyrogens are concerned, there are two different tests which are going to be used. That is said to be the rabbit test and the bacterial endotoxin test. These two tests, they will responsible for the process of ensuring or evaluation or quality control for the presence and absence of the pyrogens. Now, in case of the rabbit test, the two tests are going to be carried out. The first test, that is said to be the preliminary test, or it is also called as sham test, and the second test that is followed by the preliminary or the sham test, which is said to be the main test. Now, in order to perform this particular test, what is necessary? We need to have the rabbits of uniform weight, uniform size, uniform age. They should be maintained onto the proper diet. They should be having the proper day-night cycle, sleep, everything. They should be maintained onto the balanced diet. Their weight that should be adjusted, and their body temperature should be normal. In order to perform the preliminary test, only such rabbits are going to be used. So we need to inject the intravenous injections of the sterile paragen-free saline solution that is to be given to the animal. Note that this is the sham test, and here we are not giving directly product, but what we are giving the sterile paragen-free saline solution to exclude any animal that is showing an unusual response to the trauma or shock of the injection. That is just to ensure that whether any rabbit that is sensitive to the process of injection, or if anyone that is going to be giving the unusual response to that of the saline solution, that is rise into the body temperature, then such rabbit should not be included into the test. So any animal showing a temperature variation greater than 0.6 degrees Celsius is not used in the main test. So that is the ultimate objective why we are going to be performing the preliminary test. So just in order to ensure that there is no shock, there is no trauma which is going to be induced or inducing into the rabbits as a result of injection, and there is no unusual response, unusual rise into the body temperature by more than 0.6 degrees Celsius. Now such rabbits are going to be used. The generally 3 rabbits are selected. Now the preparation and injection of the product that is to be carried out. The product that is going to be warmed, it is dissolved or it is diluted and the duration of injection that should not be more than that of the 4 minute. The injected volume that is not less than 0.5 ml per kg and not more than 10 ml per kg of the body mass of the rabbits. The product that is going to be injected into the left ear vein of the rabbits. So determination of the initial and the maximum temperature. All the rabbits, they should have initial temperature that is from say 38 to 39.8 degrees Celsius. That is before injecting the product, and the difference into the initial T temperature that should not differ from one another by more than 1 degree Celsius. So how we can go for interpretation of the results? In case of the rabbit test, the test is carried out on the first 3 groups of the rabbits. If necessary, on further groups of 3 rabbits to a total of 4 groups, depending on the results obtained. The interval of passing and failing of the product are on the basis of the sum response temperature. So this particular table that will shows the results of the pyrogen test. Now you can see there is a number of rabbit, there is an individual rise into the temperature, and there is a temperature rise. So this is the sum temperature. This is the collective temperature, and this is the individual temperature. So in the first case, what we are doing, we are selecting the 3 rabbits to which we are giving the drug, and we are observing the individual rise into the body temperature. If the individual rise that is not more than 0.6 degrees Celsius, and if the total, or if it is more than that of the 0.6 degrees Celsius in case of individual rise, then we have to go for correlating the thing with that of the sum temperature responses. So this is the sum temperature response in all 3 rabbit. If it is more than 0.6 degrees Celsius in either one, and if we will take sum, and if the sum that is not more than 1.4 degrees Celsius, then in that case we can say that the rabbits, they will passes the test for the pyrogen. So the product that is free from that of the pyrogen. And if the first test that will fail, if the individual that is more than, and the some response that is more than 1.4 degrees Celsius, then in that case we need to take the additional 5 rabbits. So 3 plus 5, now it is to be performed onto the 8 rabbit. In case of all these 8 rabbit, if the individual temperature rise is not more than 0.6 degrees Celsius, we can say that it will passes the test. But suppose if 1 or 2 of the rabbit, they will shows rise into the body temperature more than 0.6 degrees Celsius, then we need to go for some average or the sum response of the rise into the body temperature. Now, if the sum response that is either 3.7 degrees Celsius or less than that of the 3.7 degrees Celsius, in that case we can say it will passes the test. If it is more than 3.7, then we can say it will fail the test. So it will not passes the test. If the above test that will not passes, the sample is said to be pyrogenic. That means the sample or the product that is contaminated with that of the pyrogen, and go through the sources of contamination of the pyrogen. Just we have to go for finding out what will be the source of contamination. So again, to going back, that is very, very difficult. So better to take care that not to go for contaminating at any stage. So to take care in such a way that each and every step, each and every stage, that should be free from that of the pyrogen. The next is that is the bacterial endotoxin test. This is also called as the LAL test or LAL test. So it is in order to detect the quantity of the endotoxin of the gram-negative bacterial origin. In this particular test, the reagent which is going to be used, that is said to be the LAL reagent, Limulus amebocyte lysate test, or it will contain the amebocyte lysate from the horseshoe crab. So belonging to this species Limulus polyphemus or the Tachyphyllus tridentatis. Here, this is the Limulus polyphemus or the species which is said to be the Tachyphyllus tridentatus. So this is the horseshoe crab which is obtained from the, which is having the marine origin. Now the lysates of the Limulus polyphemus that is going to be collected, and the test that is based on the primitive blood clotting mechanism of the horseshoe crab enzyme located with the crab's amoebocyte lysate blood cells. Now, the horseshoe amoebocyte blood cells, they will contain the enzymes. And when this blood that is contaminated with that of the endotoxin, that is the pyrogen, then that will lead to the process of initiation of an enzymatic coagulation cascade. So that will lead to the process of formation of the proteinaceous gel. Now, when the gel formation that will occur, it will clearly indicate that there is a presence of pyrogen, there is a presence of the endotoxin, and because of which there is a gel formation. When the gel has been formed, that means the product that is contaminated with that of the pyrogens. So the test that will fails, pyrogen present, that means the test that will fail. If the gel is not produced, then at that time there is no presence of pyrogen, that means the test it will passes. So the test performance that will depends on to the various factors, like say in order to avoid the endotoxin contamination, we need to avoid it. And before the test, we need to ensure that all the interfering factor they should not be present, like say the presence of say product or say container that will be contaminated, or the equipment that will be contaminated. Equipment they should be depyrogenated, and the sensitivity of the lysate that should be known. So how to perform this test? With the help of use of LAL reagent, we need to take the equal quantity of the solution, that is 0.1 ml of the test solution and 0.1 ml of the LAL reagent. We need to mix these two. We need to incubate these two at 37 degrees Celsius and remove the test tubes after the 1 hour and invert one smooth motion slowly. So at 180 degrees Celsius and read or observe the results whether it will passes or fails. If the gel formation had been occurred, that means the test that is passing. That means there is complete absence of the pyrogen. If the gel that is if the gel had been formed, that means there is a presence of, I want to make it clear. If the gel has been formed, it means what? It will indicate what? If the gel has been formed, it will clearly indicate that there is a presence of pyrogen, right? And thus the test that will fail means the product that will be rejected. If there is no gel formation, that means there is no presence of the pyrogen, there is no presence of the endotoxin, and the product that is free from that of the pyrogen, and thus the test that will be passes. So there are three different techniques with the help of use of the LAL reagent: the gel formation technique, the turbidity formation technique, and the color development technique. So the gel clot technique that will lead to the process of gel formation, turbidity technique that will lead to the development of turbidity, and the chromogenic technique that will lead to the development of the color. The different methods with the different steps of accuracy and the LAL test results, like say method A, method B, method C, method D, method E, and method F. Method A and B, these are related with the gel process or the gel clot method. The first, that is the limit test, and second one, that is the semi-quantitative method. The method C, that is the turbidometric kinetic method. The method D and E, these are the chromogenic kinetic methods, and the method F, that is the turbidometric endpoint methods. So the gel method, a formed gel that will give the positive result, and intact gel is not formed, that is negative result. Gel clot method, that means semi-quantitative test. The titrimetric method, the results are expressed as concentration of endotoxin as less equal or greater than lambda. Labeled lysate sensitivity is there. Method C, that is the turbidometric kinetic method. That it will depend on the onset and onset time it is needed for the reaction mixture to reach a predetermined absorbance or the rate of turbidity development. Method D is chromogenic kinetic method. It will depend on the onset time needed for the reaction mixture to reach a predetermined absorbance or the rate of color development. Method E, that is the thermogenic method. It is the endpoint method. It is the quantitative relationship between the endotoxin concentration and the quantity of chromophore released at the end of an incubation period. Quantitative method, that means it will determine the presence of quantity of the pyrogens. The turbidimetric endpoint method, again, it is the quantitative relationship between the endotoxin concentration and the turbidity, that is the absorption or the transmission of the reaction mixture at the end of an incubation period. Then the another evaluation method, which is said to be the particulate evaluation, and here we can go for observing the product against the black and white background, any container with visible particle that if seen that is going to be discarded, just a manual inspection method. We have to observe the container, glass container, transparent glass container against that of the black and white background. The black background that is to be chosen in order to detect the presence of the white particles, and the white background that is going to be selected in order to detect the presence of the black particles. This method, that is the as manual inspection, so it has its limitation. The manual limitation, like say we with the help of the naked eyes, we can go for observing the particles only more than that of the 50 micron that can be observed. Less than that of the 50 micron, that cannot be observed with the help of the normal naked eyes. The variation of the visual acuity, person to person, there are differences into the accuracy, into the acuity, into the detectability. The emotional stress may go on giving the false results. Sometimes the eye strain is there, eye fatigue is there. So all these are the limitations associated with the manual observation. There is a light obscuration method. So light obscuration method, it is used to count and measure the size of particles by means of shadow cast by the particle as it passes through a high sensitivity light beam. So as per British pharmacopoeia, Japanese pharmacopoeia, and the United States of pharmacopoeia, there are different limits for particulate matters determined by the LOPC test method. So the nominal values, these are if the volume is more than that of the 100 ml, and if it is 100 ml or less than that of the 100 ml. So the presence and absence of the greater than or equal to 10 micrometer particle. If it is more than 25 particle, that is permissible, and a particles of the size of 25 micron or more than that, the three particle, that is permissible. If the volume is less than 100 ml, then in that case, the 6000 particle of the size of greater than or equal to 10 micrometer per container. And if it is more than that of the 25 micron or equal to 25 micron, it is up to 600 particles per container. That is the limit. So we have to go for the process of filtration. In case of filtration method, the sample is to be passed through the membrane filter, and the particles that are retained onto the membrane filter, bacteria proof membrane filter, that is going to be counted. So counting 0.22 micron filter is going to be used. So we need to go for counting the particles onto the surface of the filter, and the limits has been given that the as per BPJP USP, the limit for particulate matter based on the volume, that is more than 100 ml and less than 100 ml. For the particles of the size of greater than equal to 10 micrometer, greater than equal to 25 micrometer. If the volume is more than 100 ml, then if the particle size is more than equal to 10 micrometer, the 12 number of particle per ml, that is the limit. And if it is more than 25 micron, then the 2 particle per ml, that is the limit. In case of volume less than that of the 100 ml, so per container, the 3000 particles of the size of more than that of the 10 micrometer and less than that of the 25 micrometer. And if it is more than that of the 25 micrometer, then per container, the number of particles that will be 300 per container. The last test, that is the leakage test. The negative pressure is built within an incompletely sealed ampule. The ampule, these are submerged entirely deeply into the colored solution, usually 1%